Hello friends, in this video we'll be looking at the basic unit of life on earth, that is the cell. We'll be seeing how these cells were discovered and what the different types of cells are. In the further videos we'll also be looking at the contents of a cell, that is the different organelles that can be found in a eukaryotic cell. So the cell size is very small, you all know this, and because of their small size they can be observed only with the help of a microscope. So the first compound, that is the double lens light microscopes, were constructed by the end of 16th century. And after that, by the mid 1600s, a lot of interested people were making handmade microscopes, some of which had really good quality, some of which were, having a crude quality. The discovery of cells is attributed to one such microscopist named Robert Hooke. He discovered cells in 1665. What he did is he observed a slice of cork under the microscope and he found that in the cork there were honeycomb like structures. He called these empty areas, these pores, as cells because they reminded him of the cells or the rooms in which monks used to live in a monastery. So the word cell is derived from cella, that is Latin for small room. Now, now we know that what he observed, what Robert Hooke observed, was in fact the empty cell walls of dead plant tissue. And due to this discovery, he was awarded the position of curator at the Royal Society of London, which is England's foremost scientific academy. Meanwhile, we had another scientist named Anton von Leeuwenhoek. In fact, more than a scientist, his actual earning, or he earned a living by selling clothes and buttons, but he was very interested in making microscopes and grinding lenses and constructing simple microscopes. And these microscopes that Anton von Leeuwenhoek constructed in fact had remarkable quality. So in 1674 he started observing the samples around him, environmental samples, pond samples, water samples. He, in fact, even observed his tooth samples. He took scrapings from his teeth and observed them under the microscope. And when he checked these samples under the microscope, he observed a lot of organisms that were moving back and forth. He saw a lot of structures that were moving back and forth. The best part about Anton von Leeuwenhoek is that he described these structures. So he is attributed to giving the term animalcules, which is actually the microscopic organisms that he found moving in the samples. He is also the first to describe various forms of bacteria that he obtained from the scrapings of his teeth. And not only that, whatever observations were there, all of his microscopic observations with the diagrams, he kept sending as letters to the Royal Society of London for around 50 years. But due to the skepticism at that time, the society did not believe it immediately. There were a lot of scientists who did not believe it immediately, and they sent their curator, Robert Hooke, to confirm these observations. Once Robert Hooke visited him and actually saw what he was seeing under the microscope, then there was no looking back. And then we know that many other microorganisms, many other organisms were discovered post this. So from 1674 till around 1830s, there wasn't much new discovered. There were a lot of organisms discovered, but then in 1831, we had another scientist, a Scottish botanist named Robert Brown, who discovered, or who found out, who was the first one to actually tell about the nucleus. So, nucleus had been observed in the cells before, but he was the one who first detailed it. And he detailed nucleus when he was studying about orchids. So, he said that the opaque area in the cells is what we call as a nucleus or an areola. But it wasn't until the 1830s, the late 1830s, when we came to realize the importance of cells. The widespread importance of cells was realized only when we had scientists named Matthias Schleiden and Theodor Schwann who came up with the cell theory. So in 1838, these two scientists who were working independently, Matthias Schleiden was a German lawyer turned botanist. He concluded that despite differences in the structure of various tissues, plants were all made up of cells and that the plant embryo arose from a single cell. So this is what he said. Similarly, in the same year, in 1839, we had Theodor Schwann, a German zoologist and a friend of, or a colleague of Schleiden's, who said that he published a comprehensive report on the cellular basis of animal life. So Schleiden was talking about plants, Schwann was talking about animals, but together they gave the theory a very important, a fundamental theory in science, in biology, that is called as the cell theory. We'll be looking at the cell theory shortly, but this theory was quite a revolution, and because of this theory, we again had many more discoveries that came into light. The next major discovery, or you can say observation, that happened in the branch of cytology was when we had Sutton and Boveri. These two scientists, that is Walter Sutton and Theodore Boveri, they said that chromosome is the hub of heredity. Their theory, or the chromosome theory of inheritance, said that, or it identified that chromosome is the carrier of genetic material. It also said that chromosomes are linear structures with genes that are located at specific sites called as loci along them. So this established the chromosome as the main genetic material, or the main hub of heredity. After that, we had Watson and Crick, James Watson and Francis Crick, who in the year 1953 gave us the structure of DNA. Thus, the branches of cytology, that is study of cells, microbiology, that is the study of microorganisms, and genetics were established. You can see that these three branches are actually interlinked, and these three branches were established once the structure of DNA was told to us, and after that many other discoveries were also made. Now, coming to the cell theory, which was given by Matthias Leiden and Theodor Schwann. So, the cell theory is basically a theory which is telling us, or it has three tenets. There are 3 points to this theory. These 3 points were concluded after a lot of studies on the cells of plants and animals, and they realized that they are similar structures, and that is why Matthias Schleiden and Theodor Schwann gave us the first 2 tenets of the cell theory. Now, these 2 tenets, the first tenet says that all the living organisms are made up of, are composed of one or more number of cells. So, every living organism will have cell in it. Then only you can call it as living organism. So, that was the first tenet, the first point. The second tenet of the cell theory is that cell is the basic functional unit of life. So it is the unit of structure, the unit of function of all the living beings. These were the 2 tenets of the cell theory. But Schleiden and Schwann's ideas on the origin of cells, how cells came to be, was not that insightful. They both agreed that cells could arise from non-cellular materials, and given the prominence that these two scientists had, it took many years to disprove them, to tell them that no, cells cannot arise from non-cellular material. So we had in 1855, we had Rudolf Virchow, who actually said that no, living cells cannot arise from non-cellular materials, and that is why the third tenet of the cell theory came about, that is, all cells arise from pre-existing cells by the process of cellular division. You cannot have a living cell arising from a non-living object, from a non-living entity. In fact, in 1858, Louis Pasteur disproved the theory of spontaneous generation completely, and he put all the doubts to rest by the swan neck flask experiment, which established that the cell theory is true beyond doubt. Maybe we can have that swan neck flask experiment being discussed in a different video. So these are the 3 tenets of the very important cell theory. And based on this cell theory, we now know that there are 2 types of cells. We have the eukaryotic cell and the prokaryotic cell. Eukaryotic, U is a prefix that we use for something that is true. So eukaryotic cell is having a true cell-like structure. It has a true nucleus, it has membrane-bound organelles, it is having different organelles inside it. Each of these organelles is having a particular role to play. That is what we call as a eukaryotic cell. This, what I have shown you here, is an example of a eukaryotic cell. There are different organisms which represent eukaryotes. That is the fungi, we have plants, there's the kingdom Animalia. We even have protozoans or protista, which are representing the eukaryotes. The prokaryotic cells are the first cells. Before anybody else, it was the prokaryotes that inhabited the earth. So the prokaryotic cells mainly include Monera kingdom as per the 5 kingdom classification. That includes the bacteria. So all bacteria are prokaryotic. And we also have a different domain. We now have a third domain of organisms which are called as the archaebacteria. They are also prokaryotic. So the only 2 sets of organisms which are prokaryotes, the diagram is shown over here, are Monera, that is the bacteria, eubacteria, the true bacteria, and the archaebacteria. Other than that, all the organisms are eukaryotic in nature. Of course, setting apart the viruses which are not cell-like at all. Now, these eukaryotic cells may be either unicellular or multicellular. So, the eukaryotic cells are uni or multicellular. Unicellular, like the name says, is a single celled organism. The entire organism itself is single celled. For example, the protozoans or even certain fungi, we have certain algae, they are all unicellular eukaryotes. They have only one cell in them. So the eukaryotic cell organization is quite diverse in terms of shape, in terms of organization, in terms of the number of cells, the life cycle, everything. And under eukaryotes, we have unicellular as well as multicellular organization. Multicellular is the opposite of unicellular. That means one organism is made up of multiple number of cells. For example, a fungal body, most of the fungi are multicellular. Plants are multicellular. Animals are multicellular. So these are the multicellular eukaryotes, whereas we do have unicellular eukaryotes as well. Now, these eukaryotes and prokaryotes have several differences between them, and these have been shown over here. So, we'll be studying about the different organelles that they comprise, and each of these organelles has a different role, but all of the organelles are present only in eukaryotes. As you can see over here, the eukaryotes have different organelles. They are basically membrane bound structures that are compartmentalizing the different functions in a cell. However, in a prokaryote, these organelles are absent. So all of the functions either happens in the cytoplasm, a few things happen in the plasma membrane. Similarly, we have the absence of a nucleus in case of a prokaryote, but that nucleus is present in a eukaryote. In a prokaryotic cell, the nucleus is not there, so the nuclear material, the genetic material, is spread out in the form of a nucleoid. You can even see the size. Mostly the eukaryotes are quite big in size. We do have exceptions to this. However, the prokaryotes are really, really tiny. They range from a size of zero point 0.2 to 2 micrometers in length. So they are very tiny. We have absence of different microtubules and microfilaments. These are cytoskeletal elements. So they are absent in prokaryotes, but they are present in the eukaryotes. We also have ribosome differences. Ribosome is one structure that is present in both the organisms. However, the prokaryotic ribosome is of the 70S type. S here stands for the Swedberg unit, whereas the eukaryotic ribosome is 80S type. And when we see the DNA organization, both of them have DNA as their genetic material, but in the prokaryotes, there are very few proteins that are associated with the DNA, histone-like proteins. Whereas in eukaryotes, there are many histone-like proteins which form complexes to give us the chromosome that is absent in case of prokaryotes. So these are some of the differences which we'll be looking at in detail in the upcoming videos. I hope this brief introduction and overview about a cell and how the cell was discovered was useful to all of you. I hope to see you all in the further videos as well. Thank you for all your support.